An interesting and important partnership has evolved between DNA polymerase and DNA synthesis. DNA polymerases are enzymes that can be purified and used for replication in vitro with selected DNA templates. And the ability to generate a specific primer by DNA synthesis and using the complementarity of single-stranded DNA with that primer yields a double-stranded DNA with an open 3' end and a template that allows initiation to occur and then the continuation of extension of the primer to complete a double-stranded DNA. So if we have the primer, we have the template, we have this cloud of DNA polymerase, it can move along and complete a newly synthesized, now double-stranded DNA. And this lays the basis for a process called polymerase chain reaction, for which Kary Mullis received the Nobel Prize. It requires that you have an oligonucleotide, a primer, a single-stranded DNA, and it also requires temperature-stable DNA polymerase, because otherwise DNA polymerase gets denatured at high temperature, which you have to use to separate a double-stranded DNA. So if we take a double-stranded DNA and we denature these two DNAs, we can add primers that correspond to the 3' ends of each of the longer DNAs. The primers run 5' to 3' to the 3' to 5' of the DNA. We can elongate using polymerase and get 2 copies of that DNA by copying each strand of the DNA with a primer. We can then denature for a second cycle, add the primers are already there, the polymerase is already there, and so you denature, you allow it to reanneal with the primers, and now you have 4 strands of DNA. So this is a way to amplify DNA sequences using this temperature-stable DNA polymerase that allows denaturation at high temperature, annealing at low temperature. DNA polymerase stays stable through the cycles of temperature. This allows amplifying any DNA sequence, complementary DNA derived from a sequence of messenger RNA, genomic DNA, any DNA sequence can be amplified as long as you have a primer. And that amplification is exponential. That is, first cycle you get 2 copies, and then you get twice that, 4 in the second cycle, 8 in the third, 16 in the fourth, and after 30 cycles you have exponential expansion and about 1 billion copies of the same DNA sequence. So this allows experiments with sequences that are very hard to isolate, that are very rare. And so if you explore the literature, for example, on looking at synthesis from Neanderthal DNA, that is possible because of this kind of amplification, which has to be done carefully under very controlled conditions. So what are the kinds of applications for this other than amplifying DNAs that we want to look at? Well, it turns out there are various ways that you can carry out PCR. You can do quantitative PCR, which uses a fluorescently labeled dye on the oligonucleotide to track amplification in real time. When that dye gets into double-stranded DNA, you can separate it from the primer so you can see how much has actually been amplified. You can actually use an enzyme in the first step that makes a complementary DNA from an RNA sequence. And then that RNA sequence can be amplified by using the initial RNA, the DNA oligos in the first step, and then normal PCR in each subsequent step. And there are a whole variety of variations that are all based on using an oligonucleotide, a template, and a temperature-stable DNA polymerase. But what about fragmenting DNA? We have this long, narrow molecule. Chemical and physical methods were explored over the 2 decades following the discovery of DNA structure, with no success in being able to identify ways to specifically the DNA chain. So it was not possible to break DNA into sequences that were homogeneous and could therefore be manipulated, separated, and utilized. So how can you uniquely break DNA at specific sites and do it reproducibly? So the challenge with fragmentation of DNA into smaller pieces is that you have to be able to reproducibly cut at every possible site every time. And it turns out that the solution came from a very interesting quarter, and that is bacteria. Because it turns out that bacterial restriction enzymes were discovered, and they cut at specific sequences on each strand to give products with either overlapping ends or blunt ends. And they produce the same set of fragments from a given DNA every time. So why do bacteria have these enzymes? Well, these small single-celled organisms are out in the environment. They're bathed in solutions that have lots of DNA fragments. And those DNA fragments sometimes get into the cells. And the bacteria needs a way to get rid of those fragments and protect their own DNA. We'll talk about that protection in a minute. But this is a way to protect from invasion by external DNA. And these restriction enzymes recognize specific short DNA sequences. And the strands are broken by enzymes, that is, they catalyze the hydrolysis of the sugar phosphate linkage. They recognize, again, very short, specific sequences within the DNA and cleave at that site. And you get two different types of ends. Blunt-ended, as with Haemophilus influenzae 3, this is its restriction enzyme. And BAMH1, different bacterium, different sequence, different type of cutting. So the recognition site and the type of cutting are unique to each bacterial strain. And there are literally now hundreds and hundreds of restriction enzymes are known. These are primarily bacterial, although some other single-celled organisms have these enzymes present. And these are incredibly useful in giving us the capacity to take long, complex DNAs and break them down into smaller fragments. But why is it that bacterial DNA doesn't get digested? Well, it turns out that they need these enzymes to protect them from outside DNA. But in spite of this need, they also need to protect themselves from getting their own DNA cut by these enzymes. Could you imagine a strategy that would protect the DNA of an organism from its own restriction nuclease, thereby preserving its own DNA and yet being able to digest foreign DNAs? And can you think of a way to separate the products that are produced by treating a specific DNA with a restriction enzyme? How do you separate those products, different sizes?